On a September day in 1928, Alexander Fleming returned from vacation to find that his laboratory had fallen into disrepair and his bacterial cultures had been contaminated by mold. Curiously, this mold appeared to kill Fleming's bacteria, an observation that would soon change the course of medicine. Fleming had discovered penicillin, the first true antibiotic. However, the strain of penicillium fungus that ultimately created billions of life-saving units of the antibiotic was discovered by accident on a cantaloupe in central Illinois. Accidental discoveries in the microbial realm have fueled human progress for millennia, giving rise to inventions as simple as fermented food and as complex as genetic editing tools. Throughout the course of civilization, humans have explored 1/1000% of the estimated trillion species of microbes alive on Earth, leaving the remaining microbes shrouded in darkness. These unexplored microbes are collectively referred to as microbial dark matter and may contain proteins, enzymes, small molecules, and other secrets that could be harnessed for scientific and technological advances. With revolutionary biological methods and research across the globe, an increased understanding of microbial dark matter is helping unravel not just the secrets of individual microbes, but how these microbes interact with their environments and fundamentally support all life on Earth. Microbes, a broad term encompassing all bacteria, archaea, and unicellular eukaryotes, are the most abundant and successful forms of life on Earth by almost any metric. In terms of the number of species, microbes outnumber all other organisms by several orders of magnitude. In terms of their habitats, microbes have been found at depths 10 kilometers below sea level and altitudes of over 10 kilometers, a range that exceeds any other organisms. Microbes not only survive, but thrive in boiling hot geothermal springs as well as in Arctic permafrost, often supporting their metabolism on molecules that would be toxic to other life forms. Microbes were the first life forms on Earth, and will certainly be the last. To put it simply, life on Earth would not exist without microbes, as they carry out vital functions such as nutrient cycling, photosynthesis, decomposition of dead matter, and production of metabolites valuable for other organisms. The presence or absence of microbes has a tremendous effect on other living organisms. For example, soil bacteria make nutrients, including phosphorus and sulfur, available to plants, and some are able to fix nitrogen in a form that plants can transform into the building blocks of living cells. Microbes are plentiful in the gastrointestinal tracts of many animals, including humans, where they help digest food, synthesize vitamins, and even influence our immune and nervous systems. Over the course of life on Earth, microbes have been continually evolving alongside more complex forms of life and have shaped plants and animals through their actions. Although microbes have served such a pivotal role in the development of life on Earth and the advancement of human science and technology, their actions have largely been obscured by humans' inability to see them. One of the earliest examples of humans intentionally using microbes is in the fermentation of alcoholic beverages such as beer and wine, documented over 10,000 years ago. The microscopic yeast that fermented fruits and grains had, unbeknownst to humans, come from the environment. The existence of organisms too small to see had been suggested as the cause of fermentation and contagious diseases 2,000 years ago, but it was not until Antony van Leeuwenhoek first observed rainwater under a microscope that microorganisms were truly exposed to human curiosity. In the late 17th century, van Leeuwenhoek had studied common inanimate substances under microscopes of his own creation and observed moving life forms in every sample he used, naming these new life forms animalcules. Over the next 3 centuries, the burgeoning field of microbiology gave us a greater understanding of the evolution, structure, and function of microbes and firmly established 2 new distinct domains of life. Along with an understanding of the characteristics and abundance of microbial life came the desire to learn from and apply the beneficial properties of microbes to medicine, technology, and engineering. Edward Jenner was the first to demonstrate that intentionally exposing human patients to cowpox, a mild pathogen to humans, would confer protection against smallpox, a closely related pathogen exceptionally dangerous to humans. Louis Pasteur later built on Jenner's development of vaccines by showing that exposure to artificially weakened or killed pathogens also conferred protection against the disease. The work of Jenner and Pasteur established the efficacy of vaccines in preventing diseases, applying microbes to prevent billions of deaths from contagious disease. Alexander Fleming's discovery of penicillin was only the beginning in a long list of naturally occurring antibiotics, and today, more than 3/4 of clinically available antibiotics are derived from microbes. In addition to antibiotics, microbial metabolites can often be used as medicines against chronic illnesses such as cancer, Alzheimer's, or cardiovascular disease. In the 1970s, researchers in California produced the first recombinant organism, initially by introducing genetic material from one bacterium into another, then by introducing genetic material from a frog into a bacterium. This demonstrated that microbes could be modified to synthesize small molecules, enzymes, and proteins that they normally couldn't, and thus make industrially relevant products cheaper and faster than other methods. The use of microbes for fermentation has since expanded to not only food and beverages, but also biochemical products involved in agriculture, polymer synthesis, biofuels, and energy, to name just a few examples. The tools by which microbes interact with their environment have been repurposed for human needs, including the polymerase chain reaction, restriction enzymes, and CRISPR-Cas9, methods indispensable for modern biomedicine. In the short time during which humanity has knowingly interacted with microbes, they have made an incredible impact on all aspects of our lives. Every characteristic of microbes did not arise by a single fortunate accident, but rather a series of accidents that provided an advantage and thus were passed down over millions of generations. Compared to the more easily noticeable forms of life around us, microbes have very short lifetimes, many on the order of hours. Random mutations that arise during the process of DNA replication can quickly die off or proliferate depending on their benefit to the organism's survival. Bacteria are also able to obtain new genetic information from the environment or other bacteria through transformation, transduction, or conjugation, increasing the potential diversity of genes and resulting survival advantage. Mutations in DNA are reflected in the proteins produced by microbes, which are responsible for crucial cellular functions, including nutrient digestion, energy production, adhesion and movement, and synthesis of DNA, RNA, lipids, carbohydrates, and other complex molecules. A mutation changes one or several amino acids in the protein sequence, which changes how the protein folds and how it works. Their shorter generation time is what has allowed microbes to evolve to live in such a broad range of environments. Competition for resources has driven microbes to the ends of the earth, where only those with special traits were able to survive. It is from these special traits that mankind has learned so much, and our knowledge is only scratching the surface. In the past, investigations of microbial life have relied on growing the microbes in a laboratory under specific constant conditions. This approach, however, does not work for the vast majority of microbes, with complicated growth requirements that may be dynamic or impossible to replicate in a laboratory. In some cases, it is feasible to study these microbes where they originate by growing them with carefully controlled microenvironments at the site instead of in laboratory settings. This approach, known as in situ cultivation, has already shown promise in mapping the microbiome of corals, identifying almost 2/3 of the bacteria associated with corals in hundreds of samples. In situ cultivation has also identified previously unknown terrestrial bacteria, from which thousands of metabolites have been tested as potential antibiotics. Of these metabolites, one has been shown to target the lipid membrane of bacteria rather than functional proteins, making a strain resistant to this compound much more difficult to evolve. With the use of microfluidics, hydrogels, droplet encapsulation, and other advanced techniques, in situ cultivation has been able to capture individual microbes instead of large samples. These individual microbes can then be characterized to understand their behavior or genetics and preserved for further study. While it provides the closest model of microbes' natural habitats, in situ cultivation is not a practical approach in many cases. For example, in situ cultivation at the bottom of the ocean, in a geothermal spring, or in the gastrointestinal tract of an animal would introduce too much stress on the device or make it hard to recover. Mimicking the conditions found in these environments is one possibility that has attracted attention, especially for cultivation of extremophilic archaea. The cultivation of a specific archaeon from deep marine sediment revealed the strongest evidence to date that eukaryotic organisms arose from archaea, an effort that took an entire decade of cultivation. Aside from the difficulty of replicating extreme environments in the laboratory, these methods also rely on obtaining a starter culture of the microbes from their original environment, an assumption that is far from guaranteed. Another potential method relies on sampling the air, liquids, or solids from the environment rather than the microbes themselves. The environmental samples contain nucleic acids, which can be purified and sequenced, either to determine the relative presence of specific microbial strains by 16S or RNA sequencing, or to reconstruct the entire genomes of specific microbial strains by shotgun metagenomics. With DNA sequencing becoming orders of magnitude cheaper over the last 20 years, metagenomics has become a favorite method due to the depth of information it provides. Metagenomic analysis has found approximately 8 billion different protein sequences from the biosphere, but understanding the function of many of these genes, or whether they code for proteins at all, is often impossible without analyzing the living microbes. Current trends indicate that, while metagenomics combined with machine learning may predict properties of undiscovered microbes, scaling up cultivation of newly discovered microbial species will be necessary to fully understand how the microbes' genomes determine their structure and function. In much the same way as centuries ago, investigating microbial dark matter may reveal an unexpected trove of information, since we likely won't know what we're looking for before we find it. Recent investigations into microbial dark matter have uncovered functional proteins with applications in biotechnology, relationships between microbial communities that inform the foundations of ecosystems, and the evolutionary history of life on Earth. Sediment samples collected outside of a plastic bottle recycling facility in Japan were found to contain a new species of bacteria capable of breaking down polyethylene terephthalate in plastic bottles. The genetics of this new bacteria, now known as Ideanella sacchiensis, have been used to transform other bacteria to give them the ability to break down the polymer. Enzymatic degradation of polyethylene terephthalate, and eventually many other synthetic polymers, could address the growing issue of plastic pollution and even break down polymers into their building blocks to be used again. Other enzymes could be used to break down toxic contaminants in oil spills, landfills, abandoned industrial sites, or any number of man-made areas in need of remediation. The interaction of bacteria with plant roots provides necessary signals for plants to properly develop, resist fungal and viral infections, tolerate abnormal temperatures or drought, and produce more nutritious fruit. By understanding the microbial communities responsible for plants' growth and development, we can engineer fertilizers that improve crop yields while making plants stronger, and even decrease the amount of fertilizer that needs to be applied, greatly benefiting agriculture. Microbial communities within our own gastrointestinal tracts are equally responsible for proper health, development, and disease, and manipulating those microbial communities could lead to meaningful impacts on human health and medicine. Correlating a microbe's genetic code to other information, such as where a microbe was found or when it was alive, lets us piece together the story of how the individual microbe came to be, including what evolutionary pressures it may have faced, what other microbes were around it, or how it may have impacted the living and non-living environment. By uncovering the evolutionary tree of life, we can comprehend how the variety of life around us came to be and how our actions impact it. As more and more microbial dark matter is brought to light, we will be able to apply the lessons learned from it not only to benefit humans, but also the entire planet. Thank you for watching, and as always, if you enjoyed the video, Leave a like or subscribe for more educational content. Check out some of the other videos on the channel to keep learning more about biology, chemistry, physics, history, and many more topics.